Very good evening and welcome, dear students. I cordially invite you all to 30-question discussion session. In this session, we are going to discuss about chapter number 4, Molecular Basis of Inheritance. The chapter deals with what are the molecules behind the process of inheritance and the related aspect of these molecules. Question number 1. Identify the correct order of organization of genetic material from largest to smallest. Order of arrangement from largest to smallest. The genetic material, answer is 4. Genome, chromosome, gene, nucleotide. What is genome? A complete haploid set of chromosome is called genome. That is, genome consists of many chromosomes. In the case of human being, genome consists of 23 chromosomes. Human genome consists of 23 chromosomes. Haploid chromosome number. It is the largest genetic unit. Then chromosome. Then gene. Then nucleotide. Answer is 4. Next. Which one of the following is not applicable to RNA? Which is not applicable to RNA? So RNA, it is a single-stranded molecule. Chargaff rule is not applicable to RNA. What is Chargaff rule? Chargaff rule, it is given by A by T equal to G by C equal to A plus G, that is purines, divided by T plus C, they are pyrimidines, equal to 1. That is, A by T equal to 1, G by C equal to 1, purine divided by pyrimidine is equal to 1. This is what is called Chargaff rule. Chargaff rule is applicable to double-stranded DNA. Chargaff rule is applicable to double-stranded DNA. Chargaff rule is applicable to double-stranded DNA. On 2 accounts, it is not applicable. So RNA, it is a single-stranded structure. So purine-pyrimidine ratio, number of purine divided by number of pyrimidine, will not be equal because there is no complementary pairing in RNA. There is no complementary pairing in RNA. But in DNA, there is complementary pairing. Adenine pairs with a thymine. Adenine pairs with a thymine. Cytosine pairs with a guanine. Adenine pairs with thymine. So number of adenine will be equal to number of thymine. Guanine pairs with a cytosine or cytosine pairs with a guanine. So always number of guanine will be equal to number of cytosine. So their ratio will be 1. Then number of purines will be equal to number of pyrimidines. How many purines will be there? That much pyrimidines will be there. So purine divided by pyrimidine equal to 1. This rule is called Chargaff rule, and it is applicable only to double-stranded DNA. Next question. The equivalent of structural gene. What is structural gene? Structural gene is the gene which produces mRNA, and in turn mRNA produces a polypeptide. mRNA produces a polypeptide. Structural gene corresponds to cistron. What is cistron? What is structural gene? Both of them are same. Cistron is a segment of DNA, or structural gene is a segment of DNA which are transcribed, which produce an mRNA. In turn, that mRNA produces the protein or polypeptide. mRNA produces the protein or polypeptide. mRNA produces the protein or polypeptide. So cistron. Then what is operon? What is operon? Operon is a unit which regulates gene expression. Operon is a unit which regulates gene expression. Operon consists of regulator gene, promoter gene, operator gene, and structural gene. So operon is a segment of DNA which involve or which is regulatory in function. It consists of 4 types of genes: regulatory gene, promoter gene, operator gene, and structural gene. Out of this, structural gene is called cistron. We will have a chance to discuss operon in a later question. So there we can see what is this regulator gene, what is the promoter gene, what is the operator gene, what are the structural gene. Clear? Now, what is a recon? Recon is the smallest segment of DNA which can undergo recombination. Smallest segment of DNA which can undergo recombination. That is what is called cistron. Cistron is a segment of DNA which can undergo, recon is a segment of DNA which can undergo recombination. What is muton? Muton, it is the smallest segment of DNA which can undergo mutation. A segment of DNA or smallest segment of DNA which can undergo mutation, it is called muton. Recombination is called recon. Act as a regulatory unit called operon. Next, 4. Which one of the following RNA act as structural RNA, as well as ribosome. Structural RNA, those RNA being the part of a structure, that is ribosome. The RNA which involved in the structural organization of ribosome. The RNA we know rRNA. Ribosomal RNA is involved in the structural organization of ribosome, because we studied ribosomes are made up of RNA and 80 different types of proteins. 80 different types of protein. So RNA is a ribonucleoprotein particle. Structurally consists of RNA and protein. So ribosomal RNA, they are structural RNA. Some ribosomal RNA act as ribozyme. Ribozyme means act as enzyme. RNA acting as enzyme. RNA acting as enzyme. In NCERT textbook, in translation part, that is in protein synthesis, that is, this example is given. 23srRNA. This RNA is present in the ribosome of bacteria. Since it is present in the ribosome of bacteria, it is a structural RNA. It is structural RNA. It acts as an enzyme. It acts as an enzyme for the formation of peptide bond. It acts as an enzyme for the formation of peptide bond. Generally, the enzymes which help in the formation of peptide bond is called peptidyl transferase. Peptidyl transferase, the enzyme which is responsible for the formation of peptide bond. Here 23srRNA as a ribozyme, it acts as an enzyme for the formation of formation of peptide bond. So it acts as a peptidyl transferase. So 23srRNA, it is a structural RNA as well as an enzyme which we call ribozyme also. Question number 5. Question number 5. The molecule which can act as a genetic material must fulfill the traits given below, except. Generally, if a question ends in except, that means we have to select a wrong statement regarding this. Here the question ends in except. The options given are the properties of genetic material we expect. What one property is not the property of a genetic material. The first one itself, it is wrong. It is wrong. It should be unstable is wrong. A molecule which is genetic material, one of the most important property of genetic material we expect is stability. Structural stability in terms of structure and also in terms of chemical nature. Structural organization and chemical. Structurally and chemically, the genetic molecule which act as genetic material should be stable. Others are correct. Others are correct. So we have to select a wrong statement because the question ends in except. Question number 6. An application level question. If there are if there are 999 bases in an RNA that code for a protein with a 333 amino acid at the base at 901 position is deleted such that length of the RNA becomes 998 bases. How many codons will be altered? So normally, the normal one consists of 999 bases. 999 bases. So it code 333 amino acids because 3 bases constitute a genetic code and it code an amino acid. So 3 bases are required for a codon. Each codon code an amino acid. So 999 bases, 333 amino acid. But here it says that at 901 position, the base is deleted. Base is deleted. So there is only 998 bases. 999 bases. How many amino acids? Up to here, 300 amino acids are added. From here onwards, the change will be affected because if one nitrogen base, if one base is deleted, it will cause frame shift mutation. Deletion of 1 or 2 bases. It causes frame shift mutation. What is frame shift mutation? That is a mutation which affect from the point of mutation onwards. From here onwards, it will be affected. Up to this, 300 amino acids are safely added in the amino acid polypeptide chain. 901 position, the nitrogen base is deleted. The nitrogen base is deleted. 901 position, the nitrogen base is deleted here. 901 nitrogen base is deleted. From here onwards, it affects all the rest of the amino acid. How many amino acids will be there remaining? This side remains 33 amino acid. 33 amino acid. From here onwards, it will be affected because it is a frame shift mutation. It is a frame shift mutation. Answer is 3. 33 amino acids will be affected because single base deletion result in frame shift mutation. Single base deletion result in frame shift mutation. What is frame shift mutation? Mutation affect from the point of mutation onwards. From the point of mutation onwards. Next step. So 33 is the answer, not 1. That is why they gave the option 1 above. We think that only one is deleted, 1 amino acid will be affected. No, because it results in frame shift mutation. So it affects from here onwards. Reading frame of the mRNA is altered, what we call frame shift mutation. Next, DNA fragments are, while studying the packaging of genetic material, how the DNA is packed. Negatively charged DNA wrapped on the positively charged histones. So what is the charge of DNA? The charge of DNA, it is negatively charged. It is negatively charged. Packaging of genetic material, that portion in NCERT clearly says DNA is negatively charged. Histone octamer. Histone octamer is positively charged. Histones are basic protein. They are positively charged. The DNA is wrapped around. Negatively charged DNA is wrapped around the histones. Next. Final proof for DNA as genetic material came from the experiment of. So DNA as genetic material, it was proved by who? It was proved by Alfred Hershey and Martha Chase using T2 bacteriophage. Using T2 bacteriophage, radioactive phosphorus and radioactive sulfur they used. Using T2 bacteriophage. What is a bacteriophage? A virus attacking bacteria is called a bacteriophage. T2 bacteriophage, it is a virus which attack E. coli. It is a virus which attack E. coli. So that is T2 bacteriophage. P32, it is a radioactive isotope of DNA. Radioactive isotope of phosphorus. S35, radioactive isotope of sulfur. So using these three primarily, they proved that DNA is the genetic material. Question number 9. Association of histone, that is H1, with a nucleosome indicate. So this is the H1 protein which holds the DNA together. That is, the answer is the association of histones with the DNA. It indicates that DNA are condensed, or chromatin. DNA, it is condensed to form chromatin fiber. Because the DNA is wrapped around the histone octamer and the H1 histones hold the DNA strands together without allowing to uncoil it. So it indicates condensation of DNA into chromatin fiber. Condensation of DNA into chromatin fiber. Question number 10. During DNA replication, Okazaki fragments are used. Okazaki fragments are used to elongate. So this is the Okazaki fragments. Okazaki fragments are used to elongate the lagging strand. It is used to elongate the lagging strand. These fragments are called the Okazaki fragments. But when it is lagging strand, two options are there. 2 and 4. Which one is the answer? Okazaki fragments are associated with the lagging strand. This is the continuous strand. So this answer is 4. Lagging strand away from the replication fork. This is the replication fork. This is the replication fork. So lagging strand away from the replication fork. In this direction, the lagging strand proceeds or progresses, increasing the length of the lagging strand. So, a good question, I hope. Lagging strand, everybody know. Okazaki fragments, they are fragments responsible for increasing the length of the lagging strand. But it is away from the replication fork. Replication fork is the Y-shaped junction or Y-shaped configuration formed during unwinding of DNA. Unwinding of a DNA. Next, question number 11. Select the correct match. Among the given, the correct match is the fourth one. Francois Jacob and Jack Monod, they discovered the first operon. The first operon, that is lakh operon. The first operon discovered was lakh operon. It was by Francois Jacob and Jack Monod. The organism is in E. coli. The first operon discovered was lakh operon by Francois Jacob and Jack Monod. It was discovered in E. coli. What is wrong in that? Messelson and Stahl. It is not associated with a Pisum sativum. Messelson and Stahl, they are associated with the discovery or the experimental proof for DNA replication. Semi-conservative replication of DNA. It was proposed by Watson and Crick, but proved by Matthew Messelson and Franklin Stahl. Watson and Crick could not prove the semi-conservative nature type of replication. They proposed a possibility that the DNA may replicate semi-conservatively. But full proof came from Messelson's and Stahl's work. Then, question number 12. All the following are part of an operon. The question ends in except. So we have to find out a wrong statement. The wrong one, which is not a part of operon. What is not a part of operon? Enhancer is not a part of operon. It is something related to enhancing something. But structural gene, operator gene, and promoter gene all are part of operon. All are part of operon. But enhancer is not a part of operon. Question number 13. Very carefully read the question. Base sequence of coding strand is given. If the base sequence of coding strand is given and it is asked to write the coding of mRNA, sequence of mRNA. If the coding strand is given, an mRNA sequence is asked, coding strand, sequence of coding strand, and sequence of mRNA will be same, except in the place of T in DNA, it will be U in mRNA. That is the only difference. Coding strand, you just copy down the coding strand sequence and accept it as mRNA sequence. But do one thing, at the places of T in mRNA you write U. So you write everything. So answer is C. A, G, 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 G. But you should write U. Then the other, A. Again, T comes in the coding strand. That you write U. Then C, G, C. C, G, C. A. Again, T comes, you write U. So this is the technique to write down. So you write. If the coding strand is given, but if the template strand is given, you should write the complementary strand. If the template strand is given, for example, 5 prime, A, T, G, C, A, G, C, 3 prime. If the template strand is given, coding strand I said, you write the same sequence. If the template strand is given, and if you are asked to write the mRNA, you should write the complementary one. The complementary one, 3 prime, A should be T, but mRNA it is U. T complementary is A. G, C. C, G. A, the complementary will be T, but RNA it is U. G, C. C, G. 5 prime. This is the sequence of mRNA. You write the complementary sequences. T should write A. Like that, you should write the complementary sequence. The end also mark complementary. Here it is 5 dash, so you mark 3 dash. Here it is 3 dash, you mark 5 dash. So, but coding strand is given. If coding strand is given, you write the same sequence. The only change is instead of A, write U. Here answer is 3. This is the technique of writing mRNA from the template strand. 14. Under which of the following condition there will be no change in the reading frame of the following? Change in reading frame means frame shift mutation. But frame shift mutation occurs when there is an addition or deletion of 1 or 2 nitrogen bases. If we add 3 nitrogen bases, if delete 3 nitrogen bases, it will cause mutation, but reading frame will not be altered. Reading frame will not be altered. The type of mutation in which a reading frame is altered is frame shift mutation. It is frame shift mutation. Here, answer is the third one. Answer is the third one. In the third one, 3 nitrogen bases from 7th, 8th, and 9th is deleted. If 3 bases are related, no frame shift mutation. But here only 1 is deleted. Here 2 is deleted. Here 1 is inserted. Here 1 is inserted. Insertion of 1, insertion or deletion of 1 or 2 bases result in frame shift mutation. That is alter of reading frame. Altering of reading frame. How? Insertion or deletion. Insertion or deletion of 1 or 2 nitrogen bases. It causes frame shift mutation. That is, change in reading frame. But 3 nitrogen bases are inserted or deleted. Dear students, it causes mutation, but not frame shift mutation. It may be an addition or deletion. But reading frame, it affects there only. In the other, it affects from there onwards. That is what is called frame shift mutation. Next. Purines found in both DNA and RNA. What are purines? Adenine and guanine are purines. They are double ring compounds. What are pyrimidines? What are pyrimidines? What are pyrimidines? They are single ring compound. Cytosine, thymine, and uracil. So the common purines found DNA, it is adenine and guanine. Adenine and guanine. Pyrimidines found in DNA. Pyrimidines found in DNA, they are cytosine and thymine. Pyrimidines found in DNA, cytosine and thymine. But pyrimidine found in RNA, cytosine and uracil. Pyrimidines, RNA, cytosine and uracil. DNA, cytosine and thymine. So cytosine, it is the common pyrimidine. Cytosine is the common pyrimidine found both in RNA and DNA. Cytosine is the common pyrimidine found in both RNA and DNA. But purine, both the purines, adenine and guanine, they are found both in DNA and RNA. Next, question number 16. It is a match the following. The match the following different types of genes in the lac-operon. Answer is B. Answer is B. That is I gene. I gene produces the repressor. Z gene produces the beta-galactosidase. A gene produces the trans-acetylase. Y gene produces the permease. Here, the lac-operon. This is the I gene. I gene produces the repressor. Okay, repressor. I gene produces the repressor. Z gene produces beta-galactosidase. Y gene produces permease. A gene produces trans-acetylase. These are the structural organization. There is a P gene. Now the role of I gene is clear. It produces a repressor. P gene, it is the site for binding RNA polymerase. O, it is the site for repressor. These are the structural genes. These are the structural genes. That is Z gene, ZYA. So lac-operon consists of 3 structural genes ZYA. they produces the enzyme or the protein. Now, that is the Lacoperon concept. Next, experimental verification of chromosome theory of heredity. Experimental verification of chromosomal theory of heredity. It was by which one? Not Morgan. It is Morgan. Experimental proposed. Sutton and Bovary, they proposed chromosomal theory. In biology, somebody may propose, but proof or evidence or proved by some other scientist. Here it was proposed by Sutton and Bovary, proved by providing experimental evidence, it is by Morgan. Semi-conservative replication proposed by Watson and Crick, but proved by Michelson and Stahl. This happens frequently. It is proposed by somebody, proved by other scientists. So answer is Mendel is not really concerned with this. It is with the classical genetical experiments. Question number 80. The enzyme facilitate opening of DNA during transcription. Don't commit a mistake from this. Opening of DNA during transcription. What is the enzyme involved in transcription? It is RNA polymerase. It is RNA polymerase. Don't say DNA polymerase or the enzymes involved in DNA replication. It is RNA polymerase. Question number 19. Which of the following molecules function to transfer information from nucleus to cytoplasm? Informations from nucleus to cytoplasm. Which enzyme? Which RNA? It is mRNA. The name mRNA, it is messenger RNA. It carries a message. The message for synthesis of protein. The message is carried from DNA, the genetic material, to the protein synthesizing site, that is RNA. The carrying the message, and it is called messenger RNA. Next question. Okay. tRNA, it transfer the amino acid. tRNA transfer RNA, which transfer the amino acids. So it is called transfer RNA. Formerly it was called sRNA. Recently, now it is called transfer RNA. Question number 20, with the help of a NCERT diagram, let us have a look. What is added to 3' of many eukaryotic RNAs after transcription? That is, in eukaryotes, after transcription, the RNA is subjected to some modification. This is what is called post-transcriptional events. Post-transcriptional events. 3 post-transcriptional events takes place. One is adding of a poly A tail at the 3' end. It is called polyadenylation. it is called polyadenylation. Adding of a methyl guanosine triphosphate at a 5' end. This is called capping. This is called capping. The third event is removal of introns. Unwanted bases are removed from RNA. This is called splicing. These are the three events takes place in RNA after transcription. After transcription. That is polyadenylation, otherwise called tailing, capping, and splicing. Here 3' end what is added? 3' end a tail is added at the 3' end. A cap-like structure, it is added at the 5' end. Introns are removed. Introns are removed. So this is constitute the polyadenylation or post-transcriptional events. Next, primary transcript or primary RNA transcript of the chicken ovalbumin gene. There are 7,700 nucleotides. But the mature mRNA consists of only 1,872. Primary one at the time of formation 7,700. But later the size becomes 1,872. That is said, some introns are removed. These are introns are removed. And the remaining exons are joined. That is the reason. The process is called splicing. This reduction from 7,800 to 1,872, it is due to splicing. Splicing means removal of introns. Removal of introns. Then, next. Which of the following feature would you not expect to find in heterogeneous nuclear RNA? Heterogeneous nuclear RNA is produced in eukaryotes. Heterogeneous nuclear RNA, it is produced in eukaryotes, which after modification becomes mRNA. What is modification? That is post-transcriptional changes. After post-transcriptional changes, they are transformed into mRNA. This is what is taking place in eukaryote. So what is, that is, answer is B. Polycistronic coding, it is in prokaryote. Polycistronic coding in prokaryotes. So polycistronic coding, it is absent. So these strings, intron, all these are eukaryotic structure. They are present in eukaryotes. Polycistronic coding is not, it occurs in It occurs in prokaryote. Then, which of the following is not a part of RNA processing? RNA processing is tailing, capping, joining of extrons. So this is not an RNA processing. Reverse transcription. What is reverse transcription? That is formation of RNA from DNA. The enzyme is reverse transcriptase. The enzyme is reverse transcriptase. It is not an event in post-transcriptional change. Now, 24, DNA ligase. DNA ligase is an enzyme which joins the DNA fragments. Okazaki fragments we had seen earlier in a question. They are produced in small fragments on the discontinuous strand. So these are joined together. So it is, answer is A, an enzyme that joins in normal DNA replication, which joins the fragments in normal DNA replication. Then, next. A mutation in a codon leads to the substitution of one amino acid with another. If a mutation causes substitution of one amino acid to another, it is called missense mutation. It is called missense mutation. Nonsense mutation. Nonsense mutations result in formation of a stop codon due to mutation. That is, mutation results in the formation of stop codon. Mutation results in the formation of stop codon. Then, missense mutation. Mutation results in the formation of a stop codon. If a mutation causes the formation of stop codon, it is called nonsense mutation. Frameshift mutation we studied, change in reading frame. Promoter mutation, mutation occurs at the promoter, not related to this. Next, 26 again related to the lack of. What is the inducer do? In lac-operon, what is the inducer do? You can see what is the inducer do. Inducer is the lactose. In lac-operon, inducer is the lactose. Inducer combines with the repressor. Inducer combines with the repressor, and the repressor is inactivated. Or inducer combines with the repressor, resulting in the formation of inactivate repressor. Resulting in the formation of inactivate repressor. Okay. Next. RNA primer that initiates synthesis of new strand, it is primer, initiates the. So it is called RNA primer. That is, what is RNA primer? A small segment of RNA produced at the time of DNA replication. Without this RNA primer, DNA replication cannot start. DNA replication cannot start, cannot begin. So this RNA segment is called RNA primer. 28. The reason why DNA strand is anti-parallel. What is anti-parallel? If one strand is 5 prime, 3 prime, other strand is 3 prime, 5 prime. This anti-parallel nature is due to the hydrogen bond. Hydrogen bonding between the nitrogen bases. The hydrogen bonds. Hydrogen bonding is the reason for anti-parallel nature. 29. What would be the distance between two adjacent base pair? The two adjacent base pair, the distance is 0.34 nanometer or 3.4 angstrom. 3.4, what is a nanometer? That is 10 to the power of minus 9 meter. 10 to the power of minus 9 meter. What is an angstrom? 10 to the power of minus 10 meter. Angstrom, 1 angstrom equal to 10 to the power of minus 10 meter. So to get angstrom, multiply the nanometer with a 10. Multiply the nanometer with a 10 to get angstrom. To get nanometer, divide the angstrom by 10. It is 3.4 nanometer. In DNA, the distance between two adjacent base pairs. Question number 30. It is packaging of genetic material. Diagram based question. What is A? What is B? What is C? A is DNA, which wraps around the octamer, histone octamer. B is H1 histone. C is the octamer. A, B, C in the order. A is DNA. B is H1 histone. And C is histone octamer. Histone octamer is a group of 8 histone molecules. 2 each from H2A, H2B, H3 and H4. 4 types of histones involved. 5th type is the H1. H1 is not involved in octamer. It is linking the DNA. So histone octamer consists of 4 histones, H2A, H2B, H3 and H4. This is the last question. We wind up this session of 30 question discussion of chapter 4, molecular basis of inheritance. Thank you very much, dear students. Thank you.